And I was planning to speak about plankton in turbulence, but I realized Roberto is also here and he speaks about it Friday, our common work, so I have to change the title a little bit. Biological oscillator. Nevertheless, I wanted to say something to Uriel. Happy birthday, Uriel. Whenever your birthday was. When was it?
I just died today.
I met you first time at a Nobel Symposium in 1984 here in Sweden, up north. I think the only one who was also in the room was also there, Zitoma. You remember?
That was when Leo
got the Nobel Prize for his beer. That was not so fun, but actually it was a very good meeting and very good food. And Uriel gave a fantastic lecture on the unsolved problems of turbulence. And I'm unfortunately, I think more or less all the problems are still there, despite lots of it. So Uriel, over the years since '84 got a lot of inspiration for you. I just showed 3 here. I remember with Massimo you did a very interesting work on the intermediate dissipation regime, and with Angelo Volpiani we immediately took that idea and used it in chaotic systems. Then also with Angelo and Giovanni, we started to do intermittency in shell models, and you looked me into the eyes and said, I didn't believe a word of that you have intermittency. And then I think Luca had a computer running in Nice for a month and so, and then you came and said, now I believe. And then I also remember referring to the actual discussion at lunch today in 2000 at KITP. We were sitting with Victor Jacob, and he had an idea about a fractal dimension for turbulence, 2.5. You immediately said, oh, talk to me. Because maybe what we do in shell models, and that led to some work with Victor. Indeed, there is a critical dimension in shell model around 2.5 where the flux changes its signs from forward to backward. And you have an invariant that changes continuously from helicity to entropy. And now these two prominent guys are working on this in a different context. But look at the shell model by Rahul. Good, so I changed title, but I hope you like to try to, actually we do dynamical systems in biological systems, and then in particular oscillations inside single cells, protein oscillations. I'll show you precisely how they look. And what we do is to identify the simplest feedback loop that can describe these oscillations. And what I talk about today is what's called ultradian oscillations, 2-3 hours period. And I'll show you how this oscillation can be induced by a delay. And then another interesting point I'm just working on now is synchronization with an external signal. And you get what's called Arnold tongues, a favorite topic of me. And when Arnold tongues overlap, they end in chaos, I'll show you. And then in the end, I'll show you how we couple the feedback loop and put onto a lattice to see how tissues can behave. And we also look at tissue flow. So we have a quite big group, lots of fantastic postdoc, in particular Chagor, right? Yes. Chagor.
Well, you can say Sagar because you
okay. Students, and here's some papers on this issue. Okay. But in biology, there are lots of oscillators. This is what I want to tell you. Many of you do not work in this field, so I just mention a few here. What's called cell cycle, that's cell division. And there are different phases in the cell leading to the cell divides. It can take from hours to days. And a famous, of course, oscillatory state. Then the circadian rhythms, we all know this, they 24 hours. It's what's called jet lag. All our proteins cannot follow because they're used to day and night. And so on. And people nowadays work on trying to change this. You can actually, even though we have 24 hours, you can actually...
No, it's really 24 hours only under forcing. It's not exactly for 24 hours.
That's right, that's right. That's why when you change the forcing, you can move it.
I think for people it's 23. And for mice it's 25.
That's true.
That's why you're wrong. There's actually, I think.
Or maybe the head opposite.
I think there is a spectrum. Some people have actually natural rhythm which is beyond 24. Some have maybe you. I'm sure.
Not sure.
But you're right, Itomo. If you change light, then you can move. It's quite interesting. So there's calcium oscillations inside cells. That's seconds. So there are many time scales here. And that's what I told you is what we worked on is when you do something to the cell, maybe change, damage the DNA or do inflammation and so on, you get oscillations from 2 to 5 hours. And then synchronization issues becomes very important. I'll show you that. So here is a very, very unpublished, I'm not even sure I'm allowed to show, but since you work in turbulence, I felt that no one will steal it. It's from Marcus Forward at Stanford. What single cell, just take a single human cell and stimulate it, and he can get it to oscillate around 1.5 days. Of course, it's not like a pendulum, but still, you see, it's nice oscillations. Are they chaotic? I'll come to that later. It looks a little bit. So, inside a cell, there are lots of genes and proteins and messengers and so on that interact with each other. And I come to some of the diagrams. I will not go in detail today. That's not the purpose here. But I want to tell you a little more mathematical things about a negative feedback loop. So here we have n stations. When you have an arrow, that means if you have a high concentration of this, it will promote a high concentration. If you have this dagger or whatever it is, then it means a high concentration of 2 will give a low concentration of 3. That's a negative influence, positive, negative. In order to have a negative feedback loop, you need an odd numbers of the negative ones. It's simply think about multiply minus 1. And the simplest one you can think is you have, say, some protein density. It's regulated in a negative way by itself, kind of, but with a time delay. And then you can go from a steady state, depending on the size of the time delay, into sustained oscillations. So that's the simplest negative feedback loop you can imagine. And actually there are some proteins, famous ones, that are regulated this way. Here is another famous example called the repressor introduced by Stan Leibler 10 years ago, enabled by Gilovich. So here you see you have 3 negative regulators, and actually they build this inside bacteria, and you get nice oscillations. Here again, a negative one with a self-regulation. And one we have looked at a lot is you have, say, 2 proteins and bind together in a complex. That introduces an effective delay in the regulation, and that can induce oscillations. So actually, even though we work with biology, it's rather mathematical exercise. This I just flash here. I don't have time, but that's really where we have worked the last 5 years. These are 3 very important transcription factors. This is related to cancer, this is related to inflammation, and this is to segmentation. And what we do is to write down, this is really work by my excellent postdoc Sandeep Krishna from Bangalore. And we look at how all the proteins interact. It's a puzzle, of course. And look at how the response is. And you can ask me, but what we do is try to simplify, as we learned in physics, to go to the core feedback loop that induces oscillations. And this is what I show you here. So we come down to roughly 3 proteins in this is what's called NF-kappa-B. It's NF-kappa-B, messenger inhibitor and protein inhibitor. And we reduces, I come to it, let's show one more graph of this, to 3 coupled nonlinear ODEs. And because these 2 bind in a complex, it introduces a delay, effective delay, and that's enough to induce very nice oscillations. We very often get spikes in these oscillations. And if you Google spiky oscillations, you get Sandeep's name, and we see that in almost all cases we have looked at, and actually experimentally, I'll show you, they also often see that. So let me repeat, we take the so-called genetic network, and we reduce it to a negative feedback loop that induces oscillations. So I just want to show you how people observe this. So here you have a few cells, and what's called NF-kappa-B is red, and you can see it goes into the nucleus, very nice. It's in the cytoplasm of the cell, goes into the nucleus after 1 hour roughly. It goes out, comes in again, goes out, comes in again, and so on. You directly observe with tagging a color, either red or green, and you can directly in the microscope observe these oscillations. You actually see also that you start to lose synchronization of the cell here, which is a very important issue. This kind of 2 to 5 years is oscillation is only around 5-year-old biologist has observed. So when you do this, you get this kind of, as I said, a little spiky, and this is the inhibitor is out of phase with the red curve, and they oscillate in this. These are now 5 years old, so only over half a day or so you can sustain it. But actually it fits. Okay, I'll show you actually. So we looked at, as I said, trying to, so what happens here is the following: N comes into the nucleus, it promotes a certain gene called I kappa B, and that is translated into a protein that goes back into the nucleus, binds in a complex, goes out, and then you have an external signal that degrades this complex, and free N, and it goes back in. A nice negative feedback loop. And the way we do it is usually to say that all the complexes are in equilibrium. And you can do various tricks how to reduce this. I mean, not bad tricks, I mean tricks that you can argue for because timescales, you have different timescales. And we come down to a 3-station loop with 1 negative and 2 positive regulators. And if you get the equations that looks like this, then these are 3 coupled ordinary differential equations. And it's in reduced form. I do not have too much time. You can argue for all the terms here. But we have 5 parameters here, and this is, of course, what we use quite a lot of time on from the literature to estimate these parameters. In this case, for N kappa B oscillations, we could, Sandeep found, I think, more or less all 4 of them, and one we had to guess. But we could actually, with this model, reproduce all the data with the right timescales. Here you see the spiky oscillations of N, that the inhibitor, the green, like in the experiments, is out of phase. Of course, this is deterministic model, and you, hello, this deterministic model, of course, I showed you in the data. You have very noisy data, and nowadays people are very concerned about how to kind of distinguish deterministic noise from the real regulation from stochastic noise in this kind of systems. That's a big issue.
But they're always autonomous, these.
They are always autonomous, yes, except for the fact, not in my next slide, because the external stimuli come here into this seed. It's a long story. This is an external cytokine, and you, in this case here, they put a constant, so you don't have a time dependence. However, we just did with Sandeep in the last month following calculation that you can also think that you externally force the system with a time-dependent signal. So you have an internal oscillation and an external oscillation. And this is now we are talking to by our biologists in Stanford. They're very interested in, but they didn't think about, that you can then try to synchronize an external oscillation with the internal. And you can try to get what's called anal tongues. That is, you try to synchronize the 2 oscillations. And we think, this is speculative, this could be a way to control the DNA damage and the DNA repair. And now I'll show you some very new data here. So here you have the internal oscillations as the red. This is the NF kappa B, and out of phase with the inhibitor. Then we come to the external signal. It's a square signal here. And then the modular interval. You're absolutely right. And you see here, here's the period 1 of the external. I was wrong here. There's the period 1 of the external. There are 2 spikes on the internal that's completely locked to it. And this is what we call the 2 or 1 tongue. So here's the external period. Here's the coupling between the internal and external. I'll show you one more graph of this next because it's quite interesting. If we had 3 internal oscillations on 1 external, we call it 3 over 1. And if you need 2 external to 1 internal, you call it 1 over 2. And then what you see is that this mode locking, this synchronization, gets stronger and stronger as you increase the external amplitude. The coupling gets stronger. Now they start to overlap. If we take a situation up here where the 2 tongues overlap, what kind of temporal signal do you then get? Something like this, chaotic. So the chaos comes because the system cannot figure whether it belongs to this tongue or that tongue. Or, for that matter, here comes a diagram Itamar knows very well. We worked on in Chicago many years ago. And I hope you're awake. I want to challenge you a little bit in a second. Here you see a general phase diagram of Barnum tongues. You have the external frequency, external amplitude. For each and every rational number, you have a tongue coming up. So inside the tongue, you will always have synchronization. Here from 2 to 1, 2 external to 1 internal. Here's 1 to 1, 1 external to 1 internal, and they become wider and wider where they lock to each other as you go up. Now, not so many are sleeping after lunch, that's good. So, down here we have the real axis without any mode locking. On the real axis, the rational numbers are dense. Any epsilon ball, you have infinitely many, but they have zero measure. On the real axis, the irrational has full measure. Now, what happens as you go up here, every single rational number will start to widen in a tongue. Will start to widen in a tongue. You have infinitely many rational numbers that lie dense in an epsilon ball. They all widen. Is that possible? Ladies and gentlemen?
No, they have to widen very little for those that are...
But they lie dense, right? And they all widen in size. Don't they go into each other?
They only go to each other at the one.
You're right, Ismail, yeah. Is that clear to everybody?
Yes.
It's, I believe, absolutely non-trivial. But of course, you have the rational numbers. They are, lie dense, but they have
measure zero.
Have measure zero. They have measure zero.
In the bag measure zero.
What?
In the bag measure zero.
And Lebesgue measure zero. Absolutely, yeah. Lebesgue measure zero. And therefore, even though they all widen, if the width go down fast enough, as Thomas says, they can do it. They can do it. Now let me go up to, say, this around 0.5. If I take a random point in the Lebesgue sense here, I will have a half probability to hit a tongue and half probability to hit an irrational number. Now I move, they widen, 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 widen. There's a line where if I take a random point in the Lebesgue sense here, there's probability 1 to hit a rational number and probability 0 to hit an irrational. But they are all still there. So down here, the irrational have full measure, and there are many of them, as we know. They move on lines. All the rational widens. There's a critical point where you have complementarity, they have shift role. That all the rationals has full Lebesgue measure, all the irrationals 0 Lebesgue measure, they're still all there. It's an incredibly complicated diagram, and it's true. There's a famous French mathematician called Hermann, right? Michel Hermann, where was he? Yes. Yeah, but he wrote very nice papers on this.
I mean, he has one of the best proof of the count theorem.
Okay, yeah.
Uses,
but this is not CAM, this is
I know, but it uses similar.
But it's similar. In CAM you have, resonances. It's in a very similar sense. Was it clear to everybody? So, then, if the apparently is a line where...
This is not for ladies.
There's a line where all the rationals has full measure and they still widen, then they have to overlap. They have to start to overlap. And down here, they start to overlap immediately. The very tiny tongues, but the bigger tongues overlap up here, as I indicated in different colors. And so, of course, up here, then you can see chaotic solutions where the tongues overlap. So this you will see in any system. And actually, Huygens figured that out. He had two clocks on the wall, and he figured they synchronized through the motion of the wall. So for any two oscillators, I think this is why it's very general. That's why I use time on it. This actually I worked a lot on in the 80s. Every time you have two oscillators, this will happen. And say an internal and an external, and they couple. They have to couple. And this will happen, and you see this fantastic complementarity between rational and irrational numbers. Okay, so that's what we did for this cell. The cell has oscillations, and this is a numerical calculation we do. When we put the external square wave on, actually you can also put spikes, which experimentalists tell me they like better. It works also. You get tongues. They might move a little bit. And so we have a very specific predictions now to our friends what they should look for. But this was the signal I started, I actually got last week. And here you see oscillations, but it's clearly not, certainly not the same amplitude. And what did we do 20 years ago when we saw a signal like this? Itamar, you did it more than any.
Yeah.
What did
Too much.
We try to embed it, right? We try to embed it. So here I take the signal one time, step back, two times step back, and look. 2 time steps back, 4 time steps back. This is for different series. And clearly you begin to see, it's certainly not periodic, you see some of these layered structures that we kind of like, and one should immediately calculate e to minus d2. But it's a little too early. I mean, it's a little too short for us yet. But clearly, you can see it's not a limit cycle in the hot sense. It's it has some layers, and I'm now in full collaboration with biologists. We just like a longer time series. That's all, and we can do all the tricks we know. Okay, now just to tell you a little bit more where oscillations are important. This is also very important for us. This is a mouse embryo, and in the tail of the embryo there are oscillational proteins of period 2 hours. For each period of 2 hours, a point of the spinal cord is set. This goes so much. 2 hours later, 2 hours later, 2 hours later. Actually, there's 28 here. I don't know, I forgot how many we have. 30, 40 or something. Exactly after 28 oscillations of these proteins, the spinal cord is formed and it stops. And we have, I don't know how many we have, it happened exactly the same way when we were an embryo. And people have now measured, this is also very new, they can measure oscillations of these proteins sitting there. And we are very involved in modeling this, and I'll just show you again. We write down how the proteins interact, how they bind, which gene is read, and so on and so forth. There's a student, Lykke, who does this, and you can get, depending on the parameter, stationary states, damped oscillations, it's not damped here, sustained oscillations, and so on and so forth. And of course we try to model how they stop. So after these 28 periods, it has to stop. How does it do this? It's a coupling between an oscillator and a gradient. Very elegant. People have measured in this embryo, there's a gradient of a certain protein called wind. And when the oscillator interacts, it's stimulated. It's stimulated by the wind, but when because it has a gradient and the level falls under a certain threshold, then the oscillator stops. Beautiful. And we can model this, of course. Okay, I have 5 more minutes. So now I showed you, actually we looked at this before, right? You have 2 positive and a negative that oscillates. This is what Stan Leitner did 10 years ago. 3 negative that also oscillates. It's called the repressolator. If you have 2 negative and 1 positive, that means minus times minus is plus 1. It doesn't oscillate. It's a switch. We heard about the switch this morning. It switches either from high state to a low state. Cannot oscillate. So we got this idea to take these motifs as we are long constant and put them on a lattice, a kind of condensed matter approach to tissues. So actually I'll show you in the beginning this. And if I try to put this on lattice, here are the equations. Just say, so we think about these as cells. Actually, this work with Saga involving, we think of these as cells and they interact in some way. It's a little bit abstract. So the level of a protein has a constant production and decay and some negative influence from the neighbors. So the mathematics is always like this, and it comes actually from the paper and other papers from Leibler. And now we got the idea to put it on the lattice. So you take this motif, and they all negative interactions, so you should think of this as a cell in a tissue. And so first of all, and so for each and every cell here, you have, say, a protein level that depends on time in this way. And in attack, this one is repressed by 3 neighbors and it represses 3 other neighbors. And you write the full dynamical system, and it's kind of big. And just want to tell you that this hexagonal order is very, very common in biology. It's human tissues with onion skin. So it makes sense to be on a hexagonal lattice. And so this is how the mathematics look. It's what we call multiplicative, where we multiply the influence from the neighbor. You can also add the influence from the neighbor. And of course you put this on computers and so on. And then to our big surprise, you can have a state here in this lattice where each and every cell oscillate up and down the protein density in each and every cell, completely non-phosphorated, which was a big surprise. And they're out of phase with the neighbors with 2 pi over 3 and 4 pi over 3. You have 3 phases, and each and every cell can oscillate peacefully up and down in a huge lattice without any frustration whatsoever. That was the first surprise, and I put here the numbers of the different phases. And then we began to think about condensed matter theory to do unit cells with the periodic boundary conditions. It's 3 by 3. In the periodic boundary conditions, again, you can have it in a completely unfrustrated dynamical state. Is what I showed here. You have 3 phases: red, green, blue, and with the numbers here, you see which one is what, and it comes down to a beautiful non-frustrated solution. If you do 4 by 4 with periodic boundary conditions, you actually need a 4th phase. So now the outer phase with 2 pi over 4. And we do stability theory for this and so on, understand it quite well. But if you are, maybe I'll go back a little bit, I guess also my time runs out. If you have a tissue and you have some cells here, and let's say this interact with that, it would be very natural that interact back with this cell, which we call bidirected interactions. So you have a negative this way, negative this way. And if we do this, we get local switches. It cannot oscillate at all anymore. We get local switches such that the system, here's the mathematics again. Now you see it's repressed by 6 neighbors, and it also represses its 6 neighbors. It goes back and forth. You get switches where the protein level in this cell is either in a high state or low state. This is what comes out. And then, just to finish, we talked about, okay, a natural way to look at this would be that we start with a few cells and then we make cell division and they double. And so this double and push this one up, this double and push this one there. Now this one doubles and will push there. And how does this kind of tissue grow with these kind of dynamical equations? I have a video, I hope it works. Sometimes it doesn't. It's by student Gabriel. Yeah. Okay. So here the high state is the blue and the low state is the red. And now you see, you take a random cell and it doubles and it pushes the others. Slowly, slowly. In the boundary here you have some disorder, but in the center you have a beautifully ordered ground state, dynamical ground state, where this is in a high state. I used to say that certain persons, think about an administrator, a director wants to suppress all his collaborators, all the neighbors, and that it's a very beautiful ground state, as everybody agrees actually on this. So the blue one suppresses all 6 neighbors, and you get this kind of 2 down, 1 up state, as we call it in condensed matter. So you get the ground state very nicely. If we grow it now, also with cell division on lateral growth that are full, you have cell division, all the cells on the boundary will double at the same time and look something like this. And it kind of orders, but you always get a default line. Always. You couldn't get rid of them. You see a default line going through here, and one there, and one there. And of course you have big patches with the ordered state. But somehow, when you double, I don't know how many there are here, around 100 cells, it doesn't really have time to settle down in the dynamical ground state. This of course depends on time and speed and all kinds of things, but anyhow we got this state with the default line. So the last thing, then we took these states, we took the ordered state and made mutations. That means you take, say, one that is a random one that is high and make it into a low. And what we found was that the ordered tissues are very stable to mutations. Makes kind of sense probably. Whereas if you have tissues with errors, then when we do mutations, then it gets into a disordered state. And now I make a conclusion that Eric didn't like because he was a PhD opponent on this. You forgot what you said? It may be too much conclusion, but if you have a tissue without mutations, it's much more stable to, if you have an ordered tissue, it's much more stable to mutation than if you have a tissue with various faults in. And of course you could think of that that various illnesses will develop easier in a tissue with all kinds of faults. My time is out, so I just wanted to show you that we try to do dynamical system modeling of cells, and in particular look at oscillations, and lots of the techniques we learned from UAL and others with chaos and dynamical systems will be easier. Thank you very much.
Thank you. Do you know the crystallography of the defects? Is there a coincidence lattice?
Yeah, I think it looks kind of random. That's a good point. So you think whether the default has an underlying structure, kind of?
Yeah, because you would always get grain boundaries if you grow a polycrystal.
Yes, exactly.
From a boundary.
Exactly.
You're forcing.
But very often you have these boundaries random, right?
Sure. That would be a question of rates.
Exactly. Exactly. So we have looked at, I didn't have time, we have looked at the speed of growth. Because if you have a very high speed, you get a lot of the effect.
There you go. It's geometric selection.
Structure of the oscillator equations.
Yes.
Do you guess basically, or do you have a systematic approach to finding?
We have, in which case, for the tissue or for the cell?
Both.
Oh, no, we don't guess. No, it's a very systematic approach.
So
because you know, you do a Michaelis-Menten calculation that chemists can do in sleep, right?
When you have terms in the denominator, do they come by eliminating certain reactions, or how do you?
Yes! Thank you, Axel. Fantastic question. So, you write, you do Michaelis-Menten, you say the complex is in equilibrium.
Yeah.
Then you say that the time derivative is 0, you turn it around, and you get the denominator. It's in page 20 in a chemistry book.
Oh, yeah.
We do it all the time.
Yes.
You're absolutely right. That's why the denominators come with a Hill coefficient, because depending on how many binds.
Yeah.
And
But
so
you obtained them basically from analyzing the time series.
no, that's now you get even more here, because we did indeed, from time series, predict the underlying, I didn't have time to talk about that. So you take the time series, and then you can actually predict which one enhances another one, which one decreases the other. But in general, we start with a genetic network and write up simply, ordinary differential equations. You put the complexes to 0, you invert, and you delete. Yeah, that's what everybody does in this field.
Other questions?
Thank you.